Our next speaker is Professor Onur Mutlu from the ETH the overall areas of interest are in the architecture space as well as many other spaces.
Thank you. I'll need to switch to my computer. Oh. Okay, much better. This also. Okay. Okay, great. I think Sri gave a very nice introduction. I have some introduction also, but I'm going to skip some of those slides. Now I'm going to give a little bit more deep dive into a particular problem in genome analysis. But before I start, let me give you what we think about what is our vision. Actually, genomics is part of it. Bioinformatics is a much bigger field. And I think system design for bioinformatics is really a critical problem going into the future. As Sri mentioned, it has large scientific, medical, societal, and personal implications. And I'm going to focus on genome sequence analysis. And even within genome sequence analysis, I'm going to focus on a problem called read mapping. And even within this, I think there's a lot of things that can be done, hopefully, as I will demonstrate. I have a lot of slides. I don't intend to cover all of them, but they're there for reference. And there are many bottlenecks that exist in accessing and manipulating huge amounts of genomic data. As Sri mentioned, the data is growing. It's going to grow even further. And there are many recent ideas here to accelerate read mapping. And I'm going to give you a personal journey. So where did this start? For me, at least personally. We were talking with folks at University of Washington Genome Sciences Department while I was at Microsoft Research, and they were building read mappers at the time for various reasons. And we were dreaming, basically, can we design an embedded device that can do genome analysis in real time within a minute? I think Sri's team was also looking into things like this at that time. So we have a lot of synergy in that area. And then, of course, there are many questions you can answer over here. As you can see over here, what is the likely genetic disposition of a patient to a particular drug? What disease or condition which might this particular DNA or RNA piece associated with? And then, of course, can you analyze the variations in different viruses, etc.? So there are many, many things over here that I'm not going to talk about. The applications are huge, as you can see. And clearly, there's a bright future for this. Hasunda has one of the devices. There are smaller versions of it, actually, that exist. But this is an area that is a bit hard to get into, I must say. I mean, when we first started, it's not easy to get into. So we actually more recently have written, let's say, simpler papers to help people to get into genome analysis. So if you're interested, these are some papers that I will flash, but you will have access to the slides also. And this is a more comprehensive paper that was written more recently. And we actually wrote with my student a paper for this session also, but this is a much shorter paper. If you're interested, you can take a look at it. Let me give you the bigger picture. I think this is one of the sequencing machines, Illumina, expensive. This is the next generation that Sri mentioned. Essentially, the problem we're having is we have a special purpose machine that can generate data, in this case, genomic data. It's very fast. It's very specialized. It has all these very interesting mechanisms. And then we need to analyze that data somehow. We cannot do it in the machine today. So we move a lot of data somewhere else, usually to a data center far away. And then we actually use general purpose machines that are fundamentally slower. They're not specialized for this purpose. So we have two bottlenecks, data movement bottleneck, and the other bottleneck is we use non-specialized machines for this purpose. And this talk is about overcoming both of those bottlenecks. Let's see how much we can cover. And I think to overcome these bottlenecks, we really need to design across the stack. We need to think about algorithms and accelerate them using new architectures. And we need to think across the stack to do this. So I'm going to skip some background because we don't have a lot of time, but I'm going to talk about both algorithmic and hardware acceleration, and hopefully we'll touch into some future opportunities as well. But you all have this DNA. There are actually a lot of interesting privacy issues with DNA. A lot of what we know about DNA has been done when we didn't care about privacy that much. Henrietta Lacks is one example over here. You may want to search for her. And this is the text version of this DNA. And some things we may be very interested in, like single nucleotide modifications that happen that are different between different people. They may determine your vulnerability to high blood pressure, things like that. So understanding this is important. For basically, what does this base pair correspond to, or what does the sequence of base pairs correspond to in terms of what you're looking for? So, but first, to be able to do that analysis, you first need to construct this DNA. And we have a problem. We have an immediate problem with that, basically. That machine is very powerful, but not powerful enough. Basically, we want to find the complete sequence of ACGTs in a particular portion of the DNA or the whole DNA, whole genome. Doesn't matter, depends on your analysis. The challenge is there's no machine that takes a long DNA as input. As Sri mentioned, it was 3.2 billion base pairs, right? For humans. There are much bigger DNAs also that exist. They're not humans. The machine doesn't give the complete sequence as output. Instead, all of these machines are choppers. They basically chop DNA into pieces of different sizes, but they do not tell you how they fit together. So immediately you have a puzzle coming out of this machine. And these are the choppers that you can see. There are many choppers with different properties. These are some later generation choppers, etc. These are not the latest machines, but you can see that there's a huge variety of choppers. And this is one of the nanopore machines that was mentioned earlier. And we would like to understand the capabilities of these machines. So these machines all generate data with different properties. For example, nanopore machines chop the DNA into larger pieces, longer pieces, but they're not very accurate. There are a lot of errors that happen in the machine. Some of the Illumina machines chop the DNA into much smaller pieces, like 200, 300 base pairs. And they're usually quite accurate, but there's still some errors over there. And there are other machines that we're not going to talk about that fall in between. And in the end, the goal is to solve the puzzle in read mapping at least. So if I have to illustrate it again, you have a large DNA molecule. The chopper chops it into small DNA fragments. And then these are called reads. And we want to construct what we have. That the large DNA molecule after it gets chopped. Okay, so this is really the read mapping problem that I'm going to talk about. Essentially, we want to map many short DNA fragments or reads to a known reference genome with some differences allowed. If you don't have a reference genome, you need to construct the reference genome, and read mapping can still be useful in assembling the reference genome, but I'm not going to talk about that in this particular case. So again, this picture looks like this. You chop the DNA into pieces and you map it to a reference genome to figure out where these things potentially map. Okay. Now, this problem actually is challenging for various reasons, but I will show you another example. So if you actually go around this room and sample some stuff around here and put it into one of those nanopore machines, what you would get is actually many reads that could potentially map to different reference genomes. And this is called metagenomics, let's say. You get a bunch of reads that belong to different species. And the question is, how do you identify what species they may be? So you actually have a reference database and you map these reads that come out to the reference database to figure out what you're really dealing with. So you can actually identify, for example, potential outbreaks this way, because you may actually find some unidentified DNAs also, right? But you may actually find some identified DNAs. So this is actually fascinating, I think. People are actually doing this sort of experiments that I mentioned. There are studies that have been reported where they sample stuff on the metro in New York City, for example, or the entire world, and they try to figure out what's going on. So this is very good for outbreak prediction. But of course, this is actually a much more challenging task, as you can see. So, okay, if you look at the computer science perspective, if you look at this is actually the reference genome in text file, and then you have some sequence reads in some other file. Genomics people like calling these names like FASTA or FASTQ. This is one of the reasons why it's hard to get into this area, because they have a lot of names and jargon that's not easy to get into. But basically what we're trying to find is, where is this sequence inside the reference genome? And there are different portions that may map to it, but at different probabilities, right? Okay, so essentially this is text comparison. You can do base by base comparison. I'm going to skip this. But one metric that's used to compare different pieces of text is edit distance. Edit distance is essentially the minimum number of edits needed to make the read exactly match the reference segment. So let me give you an example of this. So we have two countries over here, Netherlands and Switzerland. One of them may be the reference and the other one may be the read. And if you look at that, edit distance between them is 5. Essentially, There are two mismatches over here. There's one deletion over here that doesn't exist in Switzerland. And then there's one other mismatch, and there's another insertion over here that exists in Netherlands that doesn't exist in Switzerland. So computing this edit distance can be helpful in mapping, essentially. Okay, so let me essentially, but that edit distance computation is actually complex. So what are the challenges in read mapping? This was actually mentioned, but let me put them together a little bit. You need to find many mappings of each read. Each read may match many possible places in a reference genome. How do you find all mappings efficiently, and how do you actually build confidence that that's the right mapping? You need to tolerate small variances or errors in each read, and this may happen because of two things. One is the read that you're actually getting from the sample may not match the reference genome because there's some variation between human beings, between different species, etc. Or the machine has given you an erroneous read, right? So you actually have both of these problems. And usually, basically, you try to do this mapping with up to E errors present. And that makes it more complicated. And also, you need to do it fast, depending on the decision making. There are a lot of studies that show that if you can actually do the sequence analysis quickly, you can actually save some critically ill patients. These are actually medical studies that are reported. And they also report the cost of sequencing. Usually, some of the analyses that are very important take 2 days, 5 days. And that may not be good enough to save a critically ill patient, right? So that's why we want to accelerate it. Okay, why is read alignment slow? So this edit distance computation actually is very costly. There are dynamic programming algorithms that are traditionally being used. Recently, there are more developments in this area, which is actually fascinating. So there's a lot of algorithmic things that you can do over here that I'm going to skip because we don't have time. But take my word that it's costly. You can read about it if you're interested. And there are actually beautiful papers that are written about this and from a theory perspective as well. Okay, and if you're interested in actually read mapping techniques in general, this is an area that we've been working on for a while, together with a group of, let's say, a lot of people. We've written this overview paper in genome biology that talks about, that analyzes a lot of read mappers and different techniques that they use. And this is, of course, evolving over time. Okay, so basically, hopefully, we see where read mapping fits, right? Ideally, we would like to sequence things and then go to scientific discovery or medical advancements. But first, we need to construct the genome. And once we construct the genome, we can actually find the variants that was mentioned in Sri's talk earlier, and then actually do a lot of other studies. But we're bottleneck in this first step to begin with. Again, I'm not going to bore you with numbers, but you can see that there's a bottleneck over here. There are a lot of studies that report this bottleneck that you can read. And essentially, the picture boils down to this that I showed you. You have a very powerful machine that, and the corresponding machine that generates data, and the corresponding machine that analyzes data is very slow. Okay, so now I'll go into a little bit more detail in terms of algorithms. But again, I will not cover everything. There's a lot that's going on in this area. This is the overview paper that we talk about. And again, you can read more papers. We also have a genomics course. I will probably advertise it over here. If people are interested in getting into this area, this is live streamed, and you can actually watch whatever we've covered in the past, or you can join the next incarnation of the course and feel free to contribute. I think it's important to have special sessions and workshops like this. This is the architecture, let's say DAC design area, but it's also good to reach out to other communities. Recomb is one of the major conferences in computational biology, and we organized an acceleration workshop over there. In the future, we may organize something like this in some other, let's say, major computational biology conference. Now, okay, after all of this, let me jump into some approaches that we've been looking at. So essentially, I'm going to talk about specialized architectures, and then if we have time, we'll talk about processing in memory and storage as well. But if you look at read mapping, one of the things that you need to do is to analyze where are the cycles spent. And when we were doing these studies initially, this number was much larger. And now the number is becoming smaller. But most of the time of a read mapper is spent on sequence alignment. And we wanted to get rid of that essentially. So the overarching key idea that we first initially examined was to avoid those costly computations. And this is going to be a recurring theme. Ideally, we would like to design systems that avoid costly computations, useless computations potentially, and also useless data movement in general. So basically, we don't want to do edit distance computations or approximate string comparisons because they're costly in general. Can we actually somehow avoid that? So if you think about sequenced reads, and then you're trying to map these reads to a reference genome, some reads or some portions are too dissimilar to the reference. So ideally, we would like to identify these quickly and then filter them out. Basically, we don't want to do the comparison to the reference genome where these reads are not going to match. But For those areas where the read is similar to the reference, then we may actually want to focus our processing power over there, do the costly computations there. So this is an overarching key idea of filtering or pre-alignment filtering in general. And our first filter was actually a purely software approach that takes advantage of the structural properties of the genome, which I'm not going to talk about because we don't have time. And then we developed some metrics that I'm going to briefly mention. I think this is an area that's actually quite interesting. Can you actually develop some metrics that are much simpler to compute than edit distance, for example, and use that as a way of filtering out things? So I'm going to give you an example of this. So if you look at these two strings over here, essentially in the end, this is all text, right? We're dealing with text. We're going to talk about dealing with signals later on, perhaps, but let's deal with text right now. So one simple to compute metric is Hamming distance, right? You have a reference and then you have a read. In this case, the read exactly matches the reference. And if you compute the Hamming distance, you can figure that out. You don't need to compute edit distance. You don't need to do any sophisticated dynamic programming over here. So that's good. Now, the problem happens when you have a deletion, for example. In the read, you have a deletion that exists in the reference. Now, if you use Hamming distance over here, it's terrible because Hamming distance will tell you you have 3 matches and 5 mismatches, but you can already see that this is actually, these strings are very similar. So what we develop in this work is the idea of shifting the read. So this is non-shifted read. As you can see, you get 3 matches and 5 mismatches. But if you shift this read by 1 to the right, it looks like this. And you start the Hamming distance computation from here. And you can see that the other 4 also match. So there's only 1 mismatch over here. So the idea of shifted Hamming distance is essentially do that and shift it as many times as you think there will be errors potentially, right? And that's the idea. So we do the first comparison, Hamming distance comparison over here. So it's Hamming distance essentially XOR, as you can see. So the XOR of the first computation gives you this bit. Oh, sorry. Yeah, the first bit vector over here. The XOR of the shifted read gives you the second bit vector. And then you can add them together and then count the ones that you have. And once you count the ones, you can actually get, in this particular case, an exact distance between the 2 strings. It doesn't always work out nicely this way. That's why this is not a perfect edit distance computation, but it's a very fast way of doing this computation. So you can see that this is an algorithm that could be useful for acceleration in hardware. This is actually very easy to accelerate in hardware compared to edit distance computation. Okay, so I think I gave you the insight. You can see that this could actually be accelerated in SIMD architectures, GPUs, etc. And we looked at SIMD initially, but then we decided to build FPGA architectures that take advantage of this. And this is the gatekeeper architecture. I will not bore you with the details of the FPGA, but you can see that these bitwise operations are relatively easy to do in an FPGA. And actually, in the end, you'll be limited by the amount of data that you can bring into the FPGA. So data bandwidth becomes a bottleneck. And this was our first FPGA-based filter, and we get significant speed up in read mapping. And it's, even though it's relatively old, hopefully you should be able to download this and put it onto your FPGA and test it. So basically, one of the reasons why we actually wanted to develop this was we thought this would be a good idea to integrate with the sequencer also, so that you can do some analysis in the sequencing device itself without actually being bottlenecked by the data moment. And hopefully I'll show you at the end some industry movement that may be happening toward this direction. So you can actually do some real-time filtering and maybe on-device genome analysis going into the future. I don't think we're there yet, but that's the goal essentially. Okay, you can read more about Gatekeeper. So you can see that I wanted to cover this because it's an example of algorithm architecture co-design. There's more. I'm going to skip this one. I'm going to give you one more example, which I think the DAC community would probably appreciate. And this was based on the insights that we've developed with shifted Hamming distance. We essentially developed another filter. And if you look at this is an old graph over here. You can see that the correct alignment between 2 strings, this is one string over here, this is another string, actually 2 genomes in this particular case. Correct alignment is really a sequence of non-overlapping long matches. So ideally, you would like to look at how long is the match and then minimize the obstacles between these different long matches that happen between the 2 different strings. So if you didn't understand that, I'm going to give you another example. So basically, the key idea in this work was to reduce the approximate string matching problem to the single net routing problem in VLSI chip layout. Basically, if you think about VLSI chip layout, and if you want to route one signal from the beginning of the chip to the end of the chip over here, you want to minimize the obstacles. At least that's one way of doing the routing. So we do that. I'm going to give you an example, and we see significant speed ups compared to existing mechanisms. I'm not going to bore you with the numbers, but you can read more about it. We don't have time. So what's the key insight? Basically, you have 2 strings over here. 12, in this case, the length is 12, and the error you can tolerate is 3. Basically, what we do is we compare, we form the Hamming distance between those two strings, and main diagonal means that the strings are not shifted. And then you shift the bottom string, the read, or either one, to the left by 1, twice, and 3 times, and then compute the Hamming distance, and then shift it to the right once, twice, and 3 times, and then compute the Hamming distance. So these are actually the Hamming masks that are computed based on all different shifted versions within the error boundary. Now, if you look at this, this looks nice. 0 means there's a match. Now, you can also visualize this as this. Essentially, black parts are the obstacles that you want to avoid, and the white parts are the long matches that you actually want to maximize. So you start from the beginning. Our snake starts from the entrance, and then you route as long as possible so that you go to the longest number of whites until you reach an obstacle. Once you reach an obstacle, you decrement the count of the error because you actually spent one error budget over there. And then you route again among the longest matching whites. And then again, another obstacle, you reduce it, and then you route around the zeros, and then eventually you reach the exit over here. So this actually enables you to get a filter that doesn't perfectly compute edit distance again, but this gives you an idea of how similar the two strings are in a way that's much more easily implementable in hardware, actually in processing in memory technologies as well, because you can see the bit vectors over here. Okay, so this can actually be built on the fly. You don't need to build everything initially, so it can be memory efficient also, and we accelerate this using FPGAs and GPUs, and you can see the numbers. Okay, then we wanted to generalize some of these things. We don't want to just build filters, but we also want to do the alignment, approximate string matching in different ways, in a general way. And we built this GenAsm framework, which essentially can accelerate multiple steps of genome sequence analysis. And the interesting thing is we were actually looking at a lot of different algorithms for text search, and we found out this BiTAP algorithm that was published in 1990s. It's actually published by 2 different groups. They basically wanted to do fast text search with allowable errors. That's the title of one of the papers, actually. And we thought that this was a good fit for what we wanted to do because it uses fast and simple bitwise operations. I will not go into the details of the algorithm. You can look it up. But we modified this algorithm and extended it. And we actually also changed that so that we can support traceback, so that we can actually do edit distance computation. And co-design the hardware together with the modified algorithm. And if you do that, in the end, you end up with some sort of accelerator. In this case, you generate bit vectors for distance calculation, and then this is for traceback to make sure that you can actually compute the edit distance over here. And then this is a systolic array-based accelerator. Again, we don't have time, so I'm not going to talk about this in detail. You can find a lot more information in our paper. We did this work together with Intel, and using like state-of-the-art tools, we find out that if you integrate this on an Intel processor, you get very small area and power costs, as you can see. Okay, so this actually has multiple use cases. Pre-alignment filtering is one of them. Read alignment is another of them. And we looked at these different use cases. Again, you can read the paper for more detail. But this accelerator has significant speed up compared to existing state-of-the-art software tools, as well as hardware accelerators over here. So this sort of framework is, I think, going to be more interesting because it can do more. It's not just about pre-alignment filtering, but it can do a lot of other things at the same time. So this is, so far what we talked about is sequence-to-sequence alignment. But problems are actually getting more complex in genomics also. I'm going to skip this one, but this is an improved version of GenAsm over here. And you can actually find all of this on GitHub and look at my students' video. Problems are getting more complicated in sequence genomics. I'm going to mention one thing. So far, we want to map one sequence to another sequence, right? You have a single reference genome. But actually, this is not good in general because single reference genome cannot represent an entire species. The reference genome, maybe people are looking at representing reference genomes from different groups of people. As a result, you have actually a variation inside the reference genome itself. So how should this reference genome be represented? People have looked at, people have been, let's say, gravitating towards graph-based reference genomes. So you actually represent the reference genome as a graph. You have hops between different pieces of ACTGs, let's say. Now the problem becomes more complex because you're not mapping a read to a linear reference, but you're trying to map a read to a graph-based reference. And I think the future is actually graph-to-graph alignment, which is interesting, but We'll talk about that maybe a couple of years later. Okay, so basically we wanted to accelerate this, and we designed again another approach that's an algorithm hardware co-design to accelerate both sequence to graph mapping, but we cannot ignore sequence to sequence mapping, and we wanted to actually cover both short and long reads. Again, you can read the paper for more detail, but in the end, you need to design an accelerator. The details are too much to cover over here, but we are able to accelerate both sequence to graph mapping as well as sequence to sequence alignment with this mechanism. And you can find information about this from my students' talk at ISCA. How much time do we have left? Based on that, I'm going to decide. 6 minutes. Okay, maybe I'll talk about processing in memory. But before that, I will mention one thing that's important, I think, Sri mentioned base calling, right? The nanopore sequencer essentially measures the current, ionic current that's passing while the DNA string passes through a nanoscale hole. Now, we turn that ionic current measurements into text. That's called base calling, essentially. You call the bases that are coming. This is actually a very costly process. You use neural networks to do this, and it actually occupies a lot of the time of read mapping. And we actually have some papers that try to make it better. But I think going forward, we need to accelerate this process also. One acceleration is, of course, how do you do the base calling in a simpler way? We have this paper that's interesting, I think, multi-level, let's say, neural networks. But I think a more future-looking thing that should be happening is, why don't we just operate on raw signals as opposed to turning things into text? And this is a paper that is coming up at ISMB in a couple of weeks, which will be presented by us. I don't have time to talk about it, but I think this is actually going to be very important. Why actually can we actually operate on raw signals that are generated by the sequencing machines as opposed to converting everything to text? This actually opens up a new space of algorithms as well as hardware optimizations. Okay, maybe in the given time, I'm going to mention the processing in memory. So a lot of these techniques are actually bottlenecked by data in the end. And in the end, we don't want to move data. So we would like to be able to do some of these in memory and actually in storage. I'll go through this relatively quickly. We had a workshop on processing in memory a couple of days ago. If you're interested, you can actually look at the slides of that workshop. But if you want to actually minimize the data movement that comes out of memory or storage or sensors, all of the sequences are sensors in the end, we need to design these mapping and filtering algorithms to fit processing in memory. How do you do that? I think you can look at some papers. I will mention a couple of things over here. Actually, I'm going to skip some of these. You can certainly use emerging technologies like this, but I'm going to mention storage actually. Storage is actually quite exciting, I think, because the data sets that we're dealing with in some of these analyses are huge. It's very hard to fit some of these in memory as well. Of course, you can use algorithmic techniques to chop the data, etc. But I think storage is going to play a big role going into the future. So essentially, we move a lot of data from the storage system to the computation unit, even with all of the methods that we've discussed. So you may actually use all of these heuristics, accelerators, filters. Still, that data movement from the storage system remains, and actually it becomes a bigger bottleneck. So basically, in this work, our idea was to filter some of these reads that don't require alignment inside the storage system. So what do you filter? Hopefully you filter a lot so that you don't need to do a lot of movement of data to this part of it. So if you can actually have some accelerator in the storage system that can detect exactly matching reads, and this is actually relatively easy to do with Hamming distance, as we've discussed, you can actually filter them out. Of course, your software and mapper needs to be modified to take advantage of that as well. And also you can more easily, although not as easily as exactly matching reads, detect what is not going to match at all, let's say. Again, if you're interested, you can read the paper. There are some algorithms that you need to use over here. But in the end, we find out that even by taking advantage of limited hardware resource in the storage system, basically minimally modifying the storage system as opposed to really completely redesigning it, you get significant performance improvements. So this summarizes the results. Basically, we see significant speed ups as well as energy reduction because we eliminate this data movement from the storage. And again, the numbers are variable because it depends on what you can filter out. And this is very much depend on the application, because different use cases have different amount of exactly matching reads as well as non-matching reads. So it really depends in the end. Okay, so if you're interested in processing in memory, there's a lot more to talk about, but I don't have time. So I'll refer you to papers and talks, etc. You can also take RPIM courses. Okay, maybe I'll mention one more thing. So the processing in memory is already happening. You may say that, okay, this is future stuff. I don't care about it, but you can actually buy these chips. These are DRAM chips from UpMem. And we actually did a lot of analysis on this. And one of the workloads that we accelerated using these chips is bioinformatics, of course. So what these chips do is inside each DRAM chip, you have a processor. Okay, there you go. It's a multi-threaded processor over here next to each DRAM bank. You can actually design your algorithm such that you can do sequence analysis in this processor without moving the data out of even the peripheral logic from this bank. I think this is fascinating. If you're interested, you can actually get your hands on it. We have these real processing and memory tutorials that you can also take. And you can also find a lot of analysis. But I will mention one thing that we've done. We actually analyze a lot of sequence alignment algorithms using these processing and memory systems. This was actually recently published at Bioinformatics. You can find out more information. And once you actually redesign the algorithm to take advantage of the substrate, you get significant speed ups over CPUs and also GPUs. And I must mention that this processing in memory system has a lot of downsides today. But even then, we get significant speed ups, as you can see. So going into the future, I think processing near memory, processing in storage is going to be very, very interesting. And also processing near the sensors. Okay, so I'm going to skip all of these. Sorry, I don't have time. There should be a better way of skipping this, but there are a lot of interesting things over here. I'm going to mention future opportunities. I think there are newer genome sequencing technologies that may come up. It's good to keep an eye on that. Graph genomes are happening. Reference updates are happening. Those are also interesting. And there are a lot of work over here. And as I mentioned, raw signal analysis is going to be very interesting going into the future. Now, let me conclude. But before I conclude, I will say that there are things that are happening in industry. So even though they don't talk a whole lot about it, Illumina folks actually have FPGAs inside their sequencing machines. If you actually have some deal with them, perhaps you can use those FPGAs for variant calling, etc. NVIDIA has been doing actually interesting stuff in genomics, and they recently added these dynamic programming instructions to accelerate dynamic programming. And there are also other companies that collaborate together to do a lot of interesting analyses using GPUs. So I started with the stream earlier, right? We wanted an embedded device that can perform comprehensive genome analysis in real time, ideally within a minute. We're very far away from that today. We have a huge memory bottleneck, but we've made a lot of strides, in our opinion, collectively in the research community. So there's a lot more to do. And we haven't even talked about security and privacy, which Hasundo, I think, is going to mention also a little bit. So essentially, I think there are many future opportunities that exist in this domain, especially with new sequencing technologies and new application and use cases that are going to have a lot of impact on real lives. So there's a bright future ahead of us. I will conclude here because we don't have time. I'm happy to take any questions. Okay, there are two people. You came to the microphone first. Maybe you can go next.
Thank you. I'm Leo from University of Oregon, and actually, I'm also very interested in what you mentioned, shifting the heading distance. And I know it's trying to approximate heading distance. So, and I actually know that as you said, you have to do some work like doing ASM is for like completely the heading distance access accelerator. So I want to see your insight for like two questions actually. The first question is for like short read. How do you have an insight about what is the accuracy drop, like move from the head distance to the shifted head distance? This is my first question. And second question, and for long reads, actually, does still the shifted head distance work? Because you know it's have a larger error rate, and probably maybe when you shift a few characters, it has another error found or another insertion or deletion found. So yeah, I also want to listen to your suggestion about it.
That's a very good specific technical question. So basically, there's a drop, and we actually analyzed that in one of our papers that I didn't mention. It's magnets. If you're interested, you can take a look at it. I don't know the exact number off the top of my head, but this technique works better for short reads, as you can guess, right? Because once you go to long reads, it's very hard to do the heuristics right. That's why we actually started developing other things for long reads. But I'll refer you to the magnet paper, or we can talk offline to get the references.
Okay, thank you. And I have another question. As you mentioned, the in-memory computing is more and more important role in the for genome bioinformatics. But I feel currently the in-memory computing scheme is still facing certain data transfer. For example, for DRAM, not only the transfer between the DRAM or the main host CPU, it also have certain internal data transfer between the DRAM. So could you have some insight about what is a better way to move to deal with this data movement, and what's a more efficient way to do the calculation.
I mean, certainly processing near memory is evolving, right? So UpMem system, for example, cannot communicate between banks, and that's a major problem. But not all processing near memory systems that are proposed are like that. I think going into the future, these will evolve to minimize the data moment internally as well. But yeah, if you're looking for something off the shelf that you can use that doesn't have this internal data moment problem, it's not going to be easy to find that.
Okay, thank you.
Sure.
Brooke, I'm from Colombia. Thanks, first of all, for the presentation and also for the great online courses. My question is, so we know that in-memory computing has some problems. And have you guys explored ways of a data flow kind of acceleration of this computation so that you can read data, but you can reuse it multiple times? While this way you can avoid the problems related to in-memory computing.
So I'm not sure I understand the in-memory computing problem, but that's essentially what we do in that work that I mentioned, where we look at sequence alignment in the UpMem system.
Yeah.
You actually need to reuse the data many times inside the caching structures or the scratch pad that they have next to each bank.
Okay,
So we have definitely looked at the data reuse is still important, even if you're doing processing near memory.
okay, so what I need is accelerators with like neural, like ML kind of accelerators, which are pipeline those.
Yeah, certainly. That's why we use systolic arrays, actually. Yeah, certainly. I mean, you need to exploit the locality of the data as much as possible so that you minimize the data movement. Sure.
All right, so yeah, I was studying the read mapping algorithms a little bit, that those new algorithms these days. And then after that, I had a chance to talk to a few people from hospitals or bio startups. And it seems like that they are adopting those new technologies in a very slow rate. Yeah, so do you think there are a few challenges that are remaining to be solved, or do you think that I'm just speaking of few bad neighbors?
No, I mean, it will take time to get things adopted. Certainly, if you actually integrate your mechanisms into tool chains that are used by those folks, you can get your ideas adopted more easily. But you may not be able to examine really revolutionary ideas that way also. So I think Hasundu is going to talk a little bit about this soon. But certainly, if you use the tool chains that are used, you can do it. But if it's a complete different read mapper, and if you want to integrate it into their tool chain, then you need to do all the work and also convince those folks to use it. And those folks don't want to change anything. And this is real, unfortunately. But if you can use an FPGA to accelerate things underneath, for example, without changing the pipeline itself, it can be quite beneficial. So my suggestion is be patient, but also be cognizant of the adoption issues that may exist.
All right, thank you very much.
Sure.
Okay.